Welcome back to the channel. Iqbal Chughtai again with you people, my dear students of A levels. Today we are going to continue aerobic respiration. In our previous lecture, we discussed about glycolysis, and today's lecture we will be discussing about pyruvate oxidation, also known as link reaction, and further we are going to discuss about Krebs cycle. During glycolysis, at the end of glycolysis for one glucose molecule, we obtained two pyruvate molecules. Each pyruvate is a three carbon molecule. And with the help of special channel proteins, these pyruvate molecules enter the mitochondria, where in the matrix of mitochondria, each pyruvate molecule undergoes a link reaction. This process is also known as pyruvate oxidation. During this process, there is an oxidation process goes on and a decarboxylation process occurs as well. During decarboxylation, from each pyruvate, a carbon is released and it is released in the form of carbon dioxide, immediately followed by oxidation with the release of hydrogen that is picked by NAD molecule and it is converted to reduced NAD. This results the formation of an acetyl group. Now, a coenzyme molecule immediately joins this acetyl molecule and that transforms into acetyl coenzyme A. For one pyruvate molecule, when it proceeds in the link reaction, one carbon dioxide is lost, one reduced NAD is formed, and one acetyl coenzyme is made. But as we had two pyruvate molecules at the end of glycolysis for one glucose molecule, so the two pyruvates simultaneously proceed in the link reaction independent of each other, resulting in the formation of two reduced NAD molecules, loss of two carbon as two carbon dioxide molecules, and two acetyl coenzyme molecules produced in total. Addition of coenzyme is also resulting the same. Coenzyme here can serve two jobs. It can energize the acetyl group and it can lead the acetyl group to participate in the Krebs cycle. Because if there is no coenzyme involved, so this acetyl group can participate in any other metabolic pathway. So coenzyme ensures that this two carbon compound, acetyl or acetate, should participate in the Krebs cycle. This link reaction is occurring in the matrix of mitochondria, and acetyl group means acetyl coenzyme A will participate in the Krebs cycle that will also occur within the matrix of mitochondria. Within the matrix of mitochondria, there is a four carbon compound known as oxaloacetate that initiates this cyclic process. Acetyl coenzyme A binds with the oxaloacetate molecule and that results in the formation of a six carbon compound known as citrate. Meanwhile, coenzyme has performed its job. It leaves the cycle and moves out of the cycle. This citrate is further converted into its isomer known as isocitrate. That is also six carbon molecule. Now on the next step, decarboxylation of isocitrate will take place. That will result the loss of one carbon as carbon dioxide, immediately followed by oxidation process. Hydrogen released from the isocitrate will be picked by NAD and it will be converted into reduced NAD. After this step, now we have a five carbon compound known as alpha ketoglutarate. In the next step of this Krebs cycle, again another decarboxylation process will occur that results loss of another carbon in the form of carbon dioxide, immediately followed by an oxidation process, and hydrogen released from alpha ketoglutarate will be picked by another NAD molecule and it will also be converted into reduced NAD. Within the same step, again coenzyme makes its entry and it binds with the 4-carbon molecule, resulting after decarboxylation of alpha-ketoglutarate. And what we have at the end of this step, that is a succinyl coenzyme A. This succinyl coenzyme A is a 4-carbon molecule with 1 coenzyme attachment. In the next step, this succinyl coenzyme A will split, releasing coenzyme A, and it will be converted into succinate, that is a 4-carbon molecule. Release of coenzyme A will result the release of energy as well. During this process, when coenzyme A gets relieved from succinyl coenzyme A, energy will also be lost to the surrounding. And that energy is sufficient enough to raise an ATP molecule. Succinate is a 4-carbon molecule, and this succinate is further converted into fumarate. That is another 4-carbon molecule, but this process involves oxidation, or you can say dehydrogenation, because during this step, again, hydrogen is lost from the succinate and it turns into fumarate, and the hydrogen that is released is picked by FAD, that is flavin adenine dinucleotide, and it gets reduced to FADH2, or you can say reduced FAD. Now, fumarate will accept 1 water molecule from the surrounding, and it will be converted into another 4-carbon molecule that is known as malate. This malate will be oxidized back into oxaloacetate, and hydrogen released from malate will be picked by NAD molecule, and it will be reduced to reduced NAD molecule. And this malate, that is the 4-carbon molecule, will be reconverted back into oxaloacetate. Means the cycle started with the oxaloacetate, and that cycle ended up with the oxaloacetate, and the 2 carbon that were added in the beginning of this cycle in the form of acetyl coenzyme A, these 2 carbons were lost in the form of carbon dioxide, one when isocitrate was transformed into alpha-ketoglutarate, and the second when alpha-ketoglutarate was converted into succinyl coenzyme A. Now, for one Krebs cycle, we have a total of 3 reduced NAD molecule produced. We have got 1 reduced FAD produced. We have got 1 ATP molecule produced. But as we have 2 acetyl coenzyme A at the end of link reaction for 1 glucose molecule, so 2 acetyl coenzyme A will proceed with 2 Krebs cycles operating simultaneously. So resulting in total 6 reduced NAD molecules, 2 reduced FAD molecules, 2 ATP molecules, and all the carbon in acetyl coenzyme A, they were lost as carbon dioxide. Now proceeding with the summary of glucose molecule processing till Krebs cycle. The aerobic pathway that started with the glycolysis, continued with link reaction and Krebs cycle, what we have total so far. We have total 10 reduced NAD molecules produced, 2 from glycolysis, 2 from link reaction, and 6 from Krebs cycles. We have total of 2 reduced FAD molecules, and both reduced FAD molecules are produced during Krebs cycle. And we have total 4 ATP molecules raised so far, 2 during glycolysis and 2 during Krebs cycle. But these 10 reduced NAD molecules and 2 reduced FAD molecules will be further processed when they will be oxidized on the cristae of mitochondria during oxidative phosphorylation. So this was about Krebs cycle. Stay tuned for oxidative phosphorylation in the next lecture, and for further updates about A-level lectures, please stay tuned to this channel. Thank you very much.